Okay, thank you very much, Matthias. It's a great pleasure to be here again. So certainly I'm very, very pleased to see sunshine and warm weather here after long winter in Germany. So it's very nice to be back to Barcelona. This is a fantastic city. I really like it. So today I would like to introduce you a small story that we have developed in the lab. And so this is a study concerning MCPH1, another name called microcephaly. And I'm very pleased I saw some colleagues here and working also on microcephaly patients. I think, I hope the story will stimulate some of our discussion and hopefully also collaboration. So I think as you know, as a human being, we are very proud of us because we have a huge brain, and compared to our neighbor or very close relatives. So so this is the brain. It's on the same scale, yeah. So we have a really dramatically enlarged brain during evolution. Having said that, was a little bit cheating. Actually, we are not the human being has not the biggest brain. I think, as you know, the elephant, the whales, and even dolphin, all have either same or even bigger brains compared to the human being. But what makes the human being really, really different? Actually, if you look and in civilization quotient, and now you can appreciate the human being has a very, very big brain, and in terms of the equation compared to elephant, whales, and dolphin. Of course, after correction from body weight and all this. So therefore, human being is a very special in terms of genetic evolution and with the brain. So during evolution, I think this is just selected the brain size from our neighbors or relatives. And this is the one, and all we are very, very big compared to all the chimpanzees, bonbon, and gorilla, and all these neighbors, and we have really huge brain. This is in scale. So many people knows, and the cerebral cortex in the human being is really, really big. Actually, if you just look at the size itself, it's not something very dramatic, but the big increase actually is the surface. That make very, very different, because this is believed is the center of cognitive abilities for perception, memory, thought, and language. So that's very, very special for a human being, So, and then people believed, or we see the cerebral cortex makes us as a human being, which is different from other species or our related relatives. So, however, there are a human disease or disorders, and they have a little bit of problem with the brain size. So this is just one example. For example, this is an MRI scan, and this normal individual, you can see this is a normal cortex and the cerebellum and the brain stem, and this is, you can see also the volume, yeah. And if you look, the patients who suffer the cortical developmental disorder, and you can see here they have a much more small the cortex, yeah. So and also very, very small. This is all in the scale. So these patients suffer cortical developmental disorder, and then people believe this is also the disorder of evolutionary regression, because you basically come back to the early the relatives that the brain size get small. So I'm going to talk about today one gene or one disorder called primary microcephaly, and particularly primary microcephaly 1. And this is the patient. And this patient or disease disorder is autosomal recessive neurodevelopmental disorder. And the patients show smaller brain and growth retardation, but without very obvious neurological abnormalities. And it happened in the ones out of 30,000 or 250,000 of newborns in the Western populations. I'm not sure that's still true, but you can correct me. And the patient cells show very classical premature chromosomal condensation called PCC. And the mutations in MCPH1 caused microcephaly 1. And this is the gene I'm going to talk about. And so far, 7 genes have been mapped to a different subtype of microcephaly from 1 to 7. And all the genes until recently, I think until just 2 months ago, all are mapped. And that is a very fast developing research area. All patients show microcephaly phenotype, yeah? And perhaps some other phenotypes as well. And apparently, all these genes or molecules, they are not from the same common family, and there is no similar structure in terms of their protein structure, and they seem to not to interact directly each other. However, they all show the very similar clinical symptoms. So this is a mystery and also interesting. So basically, this is the genes. One thing, all the red seem to share some kind of a similar feature. For example, all these genes or molecules, either they are located in the centrosome or belong to a part of a centrosome or interact. So this is all located in the centrosome-related structures. So I'm going to focus on MCPH1 because this gene we started because we thought this gene may be very important for DNA damage response and also cell cycle regulation. And so we believe that perhaps this is a very interesting protein, and nobody knows the function. This is also our initial point, because my lab worked on DNA damage response for many years. So this is a scheme just to very simply or briefly summarize how this MCPH1 works. And by using patient cells or using siRNA knockdown, and the people have mapped MCPH1 at different levels of DNA damage cascade. And after DNA damage, there are 2 major DNA damage cascades. One is mediated by ATM, and another one mediated by ATR. Both are PI3 kinases. Yeah? So, an MCPH1 somehow the mutation and caused the inability of all downstream of the ATM substrate are incapable to form DNA damage-induced foci. And that's also true for ATR substrates, like RPA or RAD17 foci formation. If you're in the MCPH1 mutant cells or knockdown, and you also see compromised foci formation. And MCPH1 was discovered at the very, very beginning or reported to interact directly with the CHEK1, which are the effector of both either ATM or ATR and it's very important to control downstream of cell cycle checkpoint. So, and MCP also seem to work directly with the CDK2 cyclin A and E pathway to regulate the intra-S phase checkpoint, or through the CDK1 cyclin B1 complex to regulate the cell cycle from G2 to M. So G2 phase into mitosis. So MCPH1, I think this is very clear. MCPH1 directly interact with condensing 2, which make people believe that's responsible for chromosome, premature chromosome condensation. So the question is, what's the function of MCPH1 regulate or contribute or control the brain size through the DNA damage response, cell cycle checkpoint, or condensing 2 interaction, or centrosome functions? So it's not clear. So we took genetic approach. We tried to dissect what the function of MCPH1 finally will control the brain size. Hopefully we will learn something. So we made a knockout of the MCPH1 in the mouse. So this is the newborn brain, and this is a control animal, and this is from MCPH1 knockout. And you can see clearly the cortex is very, very small. And for example, I moved the area here, you can make a better comparison. Clearly, when you knock out MCPH1, the brain size is small, very similar to what we expected. And if you look at the cross section here, and then this is a wild type, and this is a knockout, and you can see, just look at the cortex, you can appreciate in the knockout here, they have a much thinner cortex compared to wild type controls. So what was the problem? There must be a problem during development. So we, we also looked, I put up this one because maybe it's very good to have your comment. We also tested those mice actually. Although they are small, basically they behave more or less the same. This is rota-rota test, a balance bar crossing, and then you basically found no difference between the knockout and the wild type, despite the knockout has a much smaller brain. And we also looked the learning on the memory test, for example here, and the latency and also the errors. You can see this is no difference. And also you look at this memory, and we basically found no difference of those mice compared to wild type. So despite they are small in brain, but at least the test we have performed showed no obvious neurological deficiency. So we, of course, that's not only our purpose. Our purpose is to understand what the function of the MCPH control the brain size. So we went to the developmental stage. We tried to see maybe the cells cannot proliferate properly. Actually, it's not the case. So we pass labeled the embryonic brain. This is a little bit exactly the problem. I think that's the problem, the change from my computer to here. So what I just hope later on we'll not have a bigger trouble. But just look here. What I want to tell you after BRDO labeling, yeah. So we scored all these BRDO positive cells, the past labeling that will tell you how the progenitors proliferate. So I wanted to show basically there is no difference. So no problem. They have no really proliferation problem. So it's a pity that it's lost here. And so we look at the DNA damage response. Yeah, look at ATM, look at the downstream, check 2, check 1, p53. Apparently, in those cells or progenitors, basically they have no defect in response to DNA double-strand breaks. So what could be the problem? So we performed the neurosphere formation experiment. And so you take the neuroprogenitors, you culture them to let them to form the neurosphere. Yeah. So if you look at this, and there is no difference in terms of a number of neurosphere form or the size of the neurosphere. So now called MCPH1, no problem. The neuroprogenitor can divide, can really form all the neurosphere. The big problem is the following. When we passage and dissociate the neurosphere to make a secondary neurosphere, and you can see when we make a passage 1, passage 2, and you see dramatic reduction of a number of neurosphere in the knockout, and if you look at the size, it's also very, very small. So this is one of the picture you can see from the microscope. So that strongly suggests, although MCPH1 is not essential for the proliferation, but it seems to be very important for the progenitor cell self-renewal. So then we thought about it's really true in vivo as well. So then we perform the following in vivo experiment. Before I go that, I would just give you a little bit introduction for the to refresh your neurobiology license. So this is the midbrain of the mouse here, and the forebrain and the hindbrain, and basically one can look the structure, for example, of this telecephalon. Yeah. So basically, the development of the cerebellum, cerebra or neocortex is starting from very early already. You can see the neuroepithelial cell division from E10 of mouse development, and here the newborn of the mice. So in the early case or early development, and the neuroprogenitors or stem cells, they all are located in the apical surface, basically here. Yeah. So, and then they divide, they make 2 neuroprogenitors. And with the development, and the progenitor can divide and to make 1 progenitor or 1 neuron. Or they can also make 1 progenitor, 1 intermediate progenitor that finally will give up 2 neurons. So this is how the neural stem cell, neural progenitor divide, and finally will make up all the neuron and for our brain development. So we would like to study what the function of MCPH1 in this division and in the making of the neuron. So the first experiment, we tried to map or try to measure how this progenitor stay in their stem cell progenitor compartment, or how those cells were generated in the postmitotic compartment, namely neuron. So we performed cell cycle exit assay of the progenitors. So how to do that? So we inject the BRDU into the pregnant mother, for example, in yeast 14.5, and then 24 hours later, we sacrifice and stain the brains for the antibody against the BRDU and antibody against the Ki67. That's a cell cycle marker. So using this, one can score the BRDU positive cells, Ki67 positive cells, of course, and you can also score the double positive cells. So what this means? That means those cells who are BRDU positive only, but the Ki67 negative, are those cells already exit the cell cycle. So they have incorporated once during division, that's still in the stem cell or progenitor state, and then become a postmitotic because it's no longer show the Ki67 marker. Whereas all Ki67 positive of course, regardless if they are BRDO positive or BRDO negative, they all in the cells in the cycling, namely in the progenitor compartment. Yeah, so follow this very simple formula. One can easily calculate the cell cycle exit. That means how many of those cells exit cell cycle finally. So this is the quantification. As you can see here, when we knock out the MCPH1, you see the progenitor seem to tend to exit the cell cycle. Paracel assay. So basically one can plate the progenitors in the culture dish, wait 24 hours. And the second day, basically, you follow the cells, the paired cells formed in the culture dish, which supposed to come from single progenitor. And this single progenitor may give you different composition of the daughter cells. For example, it could give two progenitor after symmetric division, or one progenitor, one neuron after a symmetric division, or neuron. So one can score this, all these pairs, by basically staining the progenitor marker nestin or stain the neuron marker beta tubulin 3. So this is just the example under the pairs, for example, progenitor, progenitor neuron, and neuron. And you can see the quantification here. In the knockout, you can see there is significant reduction of the cell division producing progenitor, but a great reduction, increase of progenitor neuron production. And no difference here. So what this means, that means when we knock out MCPH1 from the progenitors, somehow you shift the division mode from a symmetric division to asymmetric. So that's produce more this type of daughters after division. So the question is, what could be the mechanism? So we assume maybe MCPH1 is very important in this process. So this is the symmetric division. This is the apical surface. This is the ventricle. Yeah. So this is the progenitor. So usually the division is vertical, that will allow the daughter cells to inherit very important all these cell fate determinants, including part of the membrane, and that will generate two progenitors. So we questioned maybe MCPH1 deletion turned the division plane in this way. For example, that's a shift, and only one daughter inherited very important cell fate determinant, whereas another one basically did not get this. So therefore, and finally this will become a progenitor, this one become a neuron. And if you shift it even bigger, and for example, if you make a horizontal division, and only one daughter during this division inherited very important cell fate determinant, and another daughter basically did not get this. Therefore, they will become a neuron. So if that's the case, so we tested that. So we scored all the anaphase of the neuroprogenitor division from this one. So this is the division plane. This we just said that this is a vertical division, and this is a horizontal division. Yeah. So basically, we measured all the angles here. Yeah, and we come up in the following picture. And perhaps you can follow, but don't worry if you can't. And we score all these dots are individual progenitors. And with the angle from 90 to 60, 60 to 30, 30. And I just want to pay your attention. And you just look at the knockout. If you look at this population, that means the progenitor really shift their division plan. You see they have a big increase in the very, very small deviation plane. So that means when you knock out MCPH1, you really shift instead of this vertical, and they shift more and more the cells into the close to the horizontal division, but clearly that will shift to the very small angle. So that's what we have seen. So what the consequences of this? So how the cleavage plane will regulate the cell fate after the division? So we know this one and the cadherin. It's very, very important to decide for the cell fate during the division. So if in the division vertically, and the two future daughter cells will inherit the part of this membrane. This we call adherent complex, where inside you have lots of cell fate determinants. Yeah. So look, this is equal, this is unequal. Unequal means when they divide, only one daughter cells most likely inherit this part, another one miss it. So this is unequal. So if you look here and MCPH1 knockout, you can see clearly when you knock out MCPH1, somehow you shift the cell division towards unequal inheritance of these complexes. So shift mitotic or division plane, and at the same time you cause the difference in inheritance of these complexes. That's very, very important. So then the question is, then how does MCPH1 regulate mitotic or division plane? So you see all these things, you really can find out that is the problem. So one important aspect of MCPH1 and one important subcellular organelle is the spindle, right? That will regulate the division. So, and there are two very small, the subcellular organelle called centrosome. One mother centrosome and also daughter centrosome. And these centrosomes organizes spindles during cell division. So we questioned whether whether the MCPH1 is very important to influence activity of the centrosome. So first we have mapped MCPH1 to the centrosome. As you can see here, this gamma tubulin marks the centrosome, and MCPH1 GFP tag to see basically they co-localize. Clearly, MCPH1 can go to centrosome. So then what the centrosome will do to this division? So this is one scheme, the molecular cascade of centrosome function. Very, very important. So the centrosome function controls cell cycle progression. And in this case, CHEK1, as I mentioned to you, is a kinase that can phosphorylate CDC25B and to inhibit activity. Yeah? So then CDC25B activity is off. This off cannot, this is a phosphatase, yeah? And this phosphatase off cannot remove the phosphor group of CDK1. Therefore, still keep CDK1 in the inactive form. Yeah? So this is a cascade. So in this inactive form of CDK1, that will block cell transition from G2 into M phase or mitosis. Okay? So the question is whether MCPH1 will affect this. So first we looked the check one. We looked the check one by looking at the loading of check one to centrosome. So this is the control, this is the knockout, and the gamma tubulin mark the two centrosomes. And the check one, you can see this also here, yeah, and they can co-localize in the normal case. However, if you look at MCPH1 deleted neuroprogenitors, and you see you can find the centrosome marker, but you don't see the check one loading. So we quantify this. Actually, you can see in the G2 phase, and the loading of check one or localization of check one to centrosome is significantly reduced. So that means when you knock out the MCPH1 out, and the check one cannot localize to the centrosome efficiently. So the consequences, as I just showed you, is the following here. If they cannot, then this one, the phosphorylated form, may be not there anymore. Exactly the case. Look, this is MCPH1 control neurosphere and the MCPH1 knockout. If you look at the cyclin B1, there's no change. But if you look at phospho CDK1 in the knockout, you have a much less. So 39 or 40% of the progenitors contain the cells with premature CDK1 activation. So what this mean? So actually what this means when you knock out MCPH1, the check one cannot load or localize to the centrosome. So therefore, the CDC25B, the substrate, is get activated prematurely. And that will remove the inhibitory phosphor group, and this will trigger the CDK1 activity and push the cells from G2 to M prematurely. So the cells go into mitosis prematurely. It's true, and we stained phosphohistone 3 that you use this to mark the cells in the mitosis. And you can see here there are lots of positive cell numbers. So we score them. You can see in the knockout progenitors, you have a big increase of cells in the M phase after release from the cell cycle block. So that indicates eventually in the knockout MCPH1 and the cells entering mitosis earlier. So what could be the problem? So then you look at the centrosome. So we use the mature centrosome marker, ODF2, to stain these progenitors. At the same time, we measured the mitosis. Yeah. So here, symmetric localization, this marker, you can see gamma tubulin, ODF2, so they co-localize. That means the mother and the daughter centrosomes, apparently they all show mature marker. And you also find there are populations. You find two centrosomes, mother and daughter, and only one ODF2. That means the daughter centrosomes are not really mature yet. So we quantify them, and you can see here, and there's a control, this MCPH1 knockout, and you can see when we measured when the cells entering M phase, yeah, and in the knockout you see they have a much more or less cells carrying similar two centrosomes with some mature centrosome markers. So what this means? So that means there is a discoupling of the cell cycle from the centrosome cycle. So this is the model now we are thinking. So in the normal case, in the wild type, in the prophase, the mother and the daughter centrosomes, they migrate apart and to the opposite side, and then they start to organize the spindles to make ready when the cells to divide. And in the knockout, it's different. Because of the cells, so we further tested whether the CHEK1 CDC25B is really responsible for this complete change of the spindle and the cleavage change. So we use the siRNA to knock down the CHEK1. Yeah, so the same cascade. So we knock down this one to see whether we can reproduce a similar phenotype. So this is a mouse fibroblast. Yeah. So and just to show you some example, this bipolar is normal, and a multipolar and abnormal bipolar. So that is a lot of different scoring. And you can see here when we knock down CHEK1, you have a big increase of abnormal bipolar spindles. And this is a control without siRNA and siRNA luciferase. In the control, you see, you knock down CHEK1, you can reproduce the spindle defect in the fibroblast. And then we see if we knock down this one, because this is the effect, we make it, because I showed you that was prematurely activated. Now we let it die or knocked down by siRNA, and you can see here, when we make a knockdown both upstream and also this one, we can rescue. So if you block this activity, you can rescue the defect that caused by knockdown CHEK1. Yeah? And if you look at the MCPH1 knockout cells, yeah, so you can see this is, we know in the knockout cells, they have a higher abnormal spindle figures. When we knock down CDC25B, we can rescue significantly the spindle defect. So clearly that indicates CHEK1 CDC25B is very, very important for the spindle defect. So the question is, you may say, so yeah, okay, fine, but would this fibroblast really also true for the neuron? So we asked whether we can also see this in the neurodevelopment. How we do it? So we performed in utero electorportion experiment. So basically, in utero, we make electorportion of a GFP-tagged siRNA into embryonic brain. Yeah. So after certain days, one can analyze the brain development. So this is the section of the cortex. So knockdown, luciferase, CHEK1, or both. Yeah, you can always inject them into the neuron, and then basically you can score different color of the cells. And the green for the GFP, that means you have injected and integrated, transfected. And the red for Ki67, that means the cells in the cell cycle, that's progenitor, or blues DNA. Okay? So the quantification is the following. So when we made the siRNA luciferase, that is the cells make already cell cycle exit. So the cell cycle exit basically counts number of GFP positive cells only. So that means the GFP cells are those cells transfected by siRNA, yeah? But it's no longer positive for Ki67. Yeah? So, and then you divide by all the GFP positive. That means they also have a GFP Ki67 positive. So you can see there, this is a normal level. And when we knock down CHEK1, and you see they push the cells, the progenitor out of their stem cell or progenitor compartment. And when we knock down, in addition, CHEK1 and the CDC25B, we can correct this cell cycle exit, the progenitors. That's really, really, you can see that the pathway is very important for the progenitor divide and produce a neuron. So clearly we knock down the CHEK1 promotes neurogenic cell fate. Yeah? So this is a molecular pathway that really pushes them. Whether this is also true for the MCPH1. So same approach. So we also knocked down CDC25B here. We just make a CDC25B because of the downstream of CHEK1, and if we can rescue. Yes. So you can see here. So this is the brain of wild-type control, and this is the brain of MCPH1 knockout. Wild-type control, this is the basal level, yeah, luciferase, and the CDC25B, you have a little bit increase of the the neurogenic production. This is not very significant. However, as you know, as I showed before, and in MCPH1 deleted brain, you have a big increase of a cell cycle exit, namely more neuron production. Yeah? And when we knock down CDC25B, you really can see significant correction of this neuroprogenitor towards neurogenic production. So clearly, the pathway MCPH1 CDC25B is indeed control the neurogenic fate. So that's what we said. When we knock down CDC25B, we can reverse the MCPH1 phenotype. So that really comes down to the molecular pathway that's regulated by MCPH. So this is the summary. Again, this is the normal deviation, and this is MCPH1, and the premature mitotic entry, and then they have immature daughter centrosome, and that make the cells not very happy. Either they have unequal microtubule organization, and then they make the spindle change and the mitotic plane change. And in the worst case, if they cannot align properly, they will cause mitotic catastrophe and also will induce apoptosis. So we believe this premature asymmetric division increase and apoptosis both contribute to the reduction of neuroprogenitor pools finally, and will make a small brain size. So, and this is actually true when we look at the later stage of development. Yeah. So if you look in 17.5 brain, we marked with Ki67, and this control, you see lots of positive cells. That means that still lots of cells progenitor divide. And if you look at the knockout, they have a much less Ki67 positive cells. So you have a great reduction of the proliferating progenitors in the late stage. So that's really, you deplete the progenitor pools. So what could be the consequences? Why they come to the small brain? And you can see, I think just to remind you, and the brain development is in-out model. So the in-out means the first neuron produced, they stay inside, and the late stage or late newborn neurons, they make outside layer. So you just by looking at the layers, you know which ones early newborn neurons and which ones late newborn neurons. So this is the newborn, and you can see, you can mark all the layers, 1, 2, 3, 4, 5, 6. Then you can measure, or you can see how the cells, neuron produced during the later development. So this is the early newborn neuron marker called TBR1. So you can see in the control, and this layer 6, yeah? You remember, yeah? So layer 6, upward until layer 1, yeah? So you see in the knockout, you have much, much more TBR1 positive cells. That makes sense because, as I said to you before, somehow they prematurely push the knockout, push the cells, produce more neurons in the early stage. So therefore, the early newborn neurons, they have more in the knockout. And this, if you look at the CTP2, which is also intermediate, that build the layer 5 and 6, and you can see it's still high. But if you look at the late newborn neurons, and at this stage, you see they have a dramatic decrease. That's really consistent with the model that we proposed, that early switch to the neurogenic production, and then they exhausted, and the progenitor pool, therefore, they have a very, very small number of later neurons, for example, layer 2 or 3, and form. So that's therefore, it's very easy to understand how this function worked. And in the normal case, MCPH1 neuroprogenitors, they divide symmetrically, and they produce 2 neuron, progenitor, 2 neuron, 2 neuron, and later on they build up lots of neurons at the end of the development. And if you look at the knockout, so in this case, for example, they prematurely switch from progenitor division into progenitor neuron, and all these cases. Finally, you end up a very, very small number of neurons that we call a microcephaly. So finally, I would like to thank my colleagues who did most of the work. And Ralph Gruber, this PhD student, did most of the work. Zhongwei, Zhou helped him participate in the project to make in utero electorportion and also molecular analysis. Misha involved in the generation of knockout mice. Tiad was involved in the colonies. And animal facility helped us to look after and also give us technical support. Dominic, Christoph, and the histology service, and help us make lots of sections. Of course, I would like to thank the funding agencies, without which we cannot really perform all experiment. And this is a graduate school who support the student, and DFG, ISCR, DAAD, and also other agencies that support our research. So finally, I have a pleasure to introduce my colleagues, and particularly, as I mentioned, and this is the student, Ralph Gruber, who did most of the work, and the Zhongwei Zhou, and made very, very important contribution, and also Xia, the technical staff, helped us for the animal colony pedigrees. So finally, I thank you for your attention, for your time. Thank you. I stop here.